Okay, great. So you heard me verbally say these are two important disease areas of high unmet need. And today I'm going to tell you some stories around how neuroimaging biomarkers is one of many examples of the tremendous progress that will really help us get treat these disease areas with treatments on the horizon. And you know, you've heard today, the theme of today was just so well outlined, is what does it mean to be pre-competitive? And I would pose that one of the, in addition to the three things we heard about of the seminal reasons why drug approvals have happened, is really around how these pre-competitive efforts are really shaping what's needed for success. And for me personally, I was very dedicated to finding ways to pave the way for collaboration in autism, because I witnessed in my career from day one of the transformational change that happened in Alzheimer's disease to the flagship public-private partnership, ADNI, the Alzheimer's Neuroimaging Initiative. And in this disease, what's been remarkable is what started off with a focus on imaging biomarkers really ended up paving the way to identifying biomarker trajectories, modeling of disease progression, but remarkably setting a path forward for regulatory endorsement and guidance around early intervention, including prevention. So we're not there yet with the home run successful treatments that we need, but it's absolutely incredible to see the progress that's happened in Alzheimer's. And I've used this as a platform to ask, how could these kind of efforts happen in other disease areas? And before I left industry 10 years ago, this is probably my most proud achievement in my career, was writing the initial one-pager for the EU AIMS call. I had started an autism unit at Pfizer. We had 15 people, a pretty big investment, but struggling because of lack of targets and all the things we've talked about through this entire meeting. But thinking bigger, you know, how could we go beyond a single company, even though I was the largest company, research headquarters, how can we go big? And so it's incredibly rewarding for me to hear of the progress of this effort. I was involved in the initial outline of the work packages, but stepped away to do other work that you'll hear about, but so much progress. And I'll tell you why I'm excited about where this field has come based on this work. You know, EU AIMS really had incredibly ambitious goals that align with a lot of what's been discussed through this series, starting with basic science, animal models, translational approaches, and really defining new targets for intervention. So it was launched in 2012. As you know, it's now under the second phase called AIMS 2. But one of the many reasons why this European project called IMI, which there are many IMI programs, but this one really stood out because they just hit the ground running, trailblazing with 12 papers in esteemed journals, but not just publications, but what I call regulatory success in big ways. And to have the European Medicines Agency issue 5 letters of support, in this case, 3 of them are shown here in the biomarker space. The authors of this Nature Reviews paper included the EMA scientific advice reviewers, as well as the academic leads and others who led this path to achieve letters of support from the EMA based on the work that was underway. And the data hadn't even read out yet. But why is this exciting? Because the regulators can really help incentivize drug development by helping provide a catalyst for how such promising biomarkers need to be explored more and more in the future. So what is a letter of support? Well, when I came to the CPath Institute 10 years ago, there was no such thing, but our group at CPath helped work with both FDA and EMA to identify this new path where biomarker letters of support could really serve as a catalyst to encourage multiple different groups, academics, industry, and others, to evaluate in bigger ways with rigor and scientific integrity novel promising biomarkers. Why is it worth getting this letter? Well, again, it encourages identification of promising biomarkers and gets them over the hurdles of fit for purpose use. And so the more data we get, as we heard all day, the better off we're going to be to have confidence in how these biomarkers can be used to identify patients and monitor progression. So Critical Path Institute, you heard about quite a bit from Dr. Singh, who spoke in this series a few weeks ago. Dr. Singh just joined us from his academic career in autism, but it's been incredibly rewarding to work in this organization now for 9 years. It's a nonprofit in Arizona, and we are unique in that we lead public-private partnerships that achieve regulatory milestones that cannot be achieved on their own. I'm going to be showcasing a couple of examples to you today, polycystic kidney disease and Parkinson's, because they have some great successes in imaging biomarkers. But letters of support is something we are really good at, and as well as full qualification and regulatory endorsements for a variety of tools. Because as we just heard from the Roche team, we really want to compete on the chemical matter itself and not on the drug development tools that are needed to get these promising treatments to patients. So when I worked at Pfizer, we had set up this autism unit. The real catalyst for why everybody got behind this at Pfizer and beyond was this whole idea that the advances in genetics are really paving the way. And even the first speakers of this series, Dr. Buxbaum and Dr. Geschwin, really helped give talks to our leadership to say now's the right time. This initiative, we outlined a paper that looked a lot like a paper that came out many years ago, sponsored by Society for Neuroscience, and one of their sessions around how do we go from an incredibly heterogeneous disease and take advantage of viable therapeutic targets based on genetics, and all of you know that. So thanks to paving this path, hearing from this meeting series, clearly it's come so far. We're just really not where we need to be in terms of getting incentives for companies to want to advance gene-based therapies in bigger ways. So how do we do that? So I'm going to share with you now my current role, and my fast forward to today, and I lead a worldwide Parkinsons Consortium. And I think you're going to be really intrigued to hear today what I have to share with you about Parkinson's, because even though, again, these are different disease areas with different experts working across the globe, they have incredibly amazing commonalities. In Parkinson's, like autism, the genetics is really paving the path in terms of novel genes that have been identified that occur in small sets of subjects, but are completely elucidating exciting, promising pathways and targets. And I won't go through these one-on-one, but what's been fun for me about listening to this series every Sunday is realizing that every week I would come away with a checkbox of, wow, look how much autism and Parkinson's have in common. Unsustainable rates of disease increase, shared mechanisms. It's incredible to see the progress in the link in the microbiome in autism. Well, that's happening as well in Parkinson's disease in big ways. The urgent need for biomarkers, that's what was so exciting when John approached me about giving this talk. And the heterogeneity is a huge gap that we need to try to figure out and understand how we can enable precision medicine approaches, combination therapies, and I'll end with an exciting project I'm leading now in the digital technologies arena. So this consortium we founded 5 years ago with generous funding from Parkinsons UK in London, and they were funding several cohorts and wanted to go beyond publications, share that data with us and pull that data together. So in the 5 years since we launched, we have about 11,000 people with Parkinson's integrated into this database. I'll tell you about the database in just a minute, but that data has been used to achieve some exciting regulatory successes. And then I'll end and tell you about our digital team, but it's incredibly rewarding. We have 7 charity groups, we have people with the disease, we have 13 companies, 25 academic leaders, and FDA and EMA are actually part of our teams. So we set out in the very beginning to do what I keep hearing so many speakers at this conference talk about. Wouldn't it be incredible if we could put all the data from autism in one place? And just today, I've heard this is really happening. So how incredible is this? But we at CPath do the hard part, and FDA funds us to take existing data, fully standardize that data, and integrate it into a unified clinical database. But that data then becomes the substrate to develop these tools. And when we started this project in Parkinson's, we realized when everyone sat together in person in a meeting in London years ago, if we tried to go out and get every study at every stage of the disease, we'd be doing this for 10 years. So we very carefully selected a defined target population for acquisition of the data for a couple of reasons. This stage we call early motor Parkinson's, right after diagnosis within the first few years, was the area that companies were working on to try to intervene early. There's a lot of data out there. But more importantly, people with the disease had expressed that their dream would be to have a drug that would halt disease progression early, soon after diagnosis, when they could still work and have a life. So we went out and aggressively spent many nights, days, weekends to working hard to do all the legal agreements and bring this data together. At this point, we have 5 observational cohorts from multi-site studies around the world and 13 clinical trials. Most of the trials we have failed to reach their primary efficacy endpoint, but what a remarkable opportunities to learn from these studies, many of which took place years and years ago. There's so much to be learned. So the next few slides I'll just share with you. In the time since we've received the data, we have some great successes to share. One has to do with imaging biomarker success with the regulatory agencies, and the other has to do with genetics. So I'll walk you through those briefly. Here's the references if you'd like to take a look. And when I left industry, one of the hot therapeutic targets that was based on genetics findings is this gene LRRK2. It's probably one of the most common risk genes for Parkinson's, familial Parkinson's, although overall the incidence is quite low in idiopathic. But it's been studied quite a bit in terms of mechanistically, how does this gene link from the pathobiology to disease onset and risk. So our clinical team, led by the Merck team, as well as our modeling team, worked very diligently with our database to show that the actual rate of disease progression of subjects with LRRK2 was actually statistically significantly slower than idiopathic. But this was remarkable because most of the companies went all in on this target thinking that they could do a proof of concept study with fewer subjects and have a home run that then they could bring into a larger population. So this is really important data, because if you don't design the study right up front, you're not going to be successful. So the vision in Parkinson's, I'm sure this slide is exactly where we want to be in autism. We need to go from having an incredibly heterogeneous condition where each patient is going to look different clinically, biologically, genetically, and to try to get to a point where we are in oncology, where we define the target based on the phenotype and endophenotype of the patient. So how do we get there? So our Parkinsons Consortium, we're really proud, worked for several years to bring forward a biomarker very similar to the path that Jamie McPartland outlined this morning, forward to the regulatory agencies for endorsement. And we did this with data from our database, and it turns out that there's a significant number of subjects that went into clinical trials that turned out to have imaging signatures that looked like normal brain, even though clinically they look like Parkinson's. So the dopamine transporter imaging has been studied for many years. It's used as an ancillary clinical test to help differentiate between disorders, but it wasn't until we brought all this data forward that we received a letter of support from both FDA and EMA and a full qualification decision for the use of dopamine transporter for trials for early stage disease, because what we found was a significant number of patients, up to 15% of people in trials, ended up not having a biomarker deficit, and those people did not progress and they were unlikely to have Parkinson's. So this is this patient stratification approach that we really want to help enable for autism. We heard about this today. How do you identify the right subjects before you put them in a trial? A couple of other successes I'll share. On the imaging front, I was able to lead an effort in Alzheimer's to qualify hippocampal volume. Low baseline hippocampal volume is an enrichment tool for early stage Alzheimer's trials years ago. But the one that I think you're going to really like to hear about and how this is relevant to autism is polycystic kidney disease. This is, of course, not a brain disorder, but the path we took was pretty remarkable. The CPath Institute founded years ago the Polycystic Kidney Disease Consortium, fully funded by the PKD Foundation, at a time where there was literally no companies working on drug development for PKD. It's a rare autosomal dominant condition, but no incentives to work because the existing measures were not really viable to bring forward to promising targets in any kind of way that would help enable advancing novel drug development targets forward. So this consortium brought data together to look at the role of total kidney volume as a promising imaging biomarker to identify first as a prognostic marker, but then later, over time, the FDA is now accepting this total kidney volume as a reasonably likely surrogate endpoint. This is a lot of work that went into bringing observational cohort data together, integrating that data, producing a joint model comparing how TKV compared with EGFR, the traditional measures. And it's very exciting because over the years that this took place, this project went from having literally no industry involvement to now more than 10 companies pursuing therapeutic candidates for PKD. Because now, based on this regulatory path, there is a viable biomarker that people can utilize to advance compounds and know that we're going to treat patients at a time when their kidneys are still intact. So this is an example of success where really we need to incentivize industry. It's incredibly exciting to see the progress of the Roche team, but very few companies, as you know, are really investing big in autism. So a couple more slides, how did we get here? So this is kind of our framework, a diagram of how we bring data together at CPath across all the disease areas we work on. We bring in longitudinal data, academic, and clinical trial data is incredibly important to learn from. We bring that data together. We have an incredibly talented data and quantitative medicine team and use this to model disease progression over a variety of different diseases. But the last thing I'll tell you is the most exciting project I'm leading now. We just had this paper published just 2 weeks ago, just 2 weeks ago tomorrow, in special open access issue of digital biomarkers. And this was a tremendous effort of a worldwide group of people who worked together over a year or so to pave the path of how to advance digital technologies in Parkinson's for efficient use in trials. As you know, these technologies hold a lot of promise. They can be done in the home. They're very relevant to what's important to patients in terms of clinical meaningfulness. In light of COVID, there's a huge wealth of progress that's occurred. We're not where we need to be in terms of legal issues of sharing, how to deal with such high volumes of data, and how to have confidence in it, but we're making great progress. And this paper outlines the path of how FDA and EMA became part of the teams to enable what success looks like so that we can all have confidence in how they can be used efficiently. The last thing I'll tell you is we were working closely with some really talented people in the Netherlands. And they published this paper that I just wanted to bring to your attention. I encourage you to contact these folks to learn more, but it's a pretty small N, but the folks at Center for Human Drug Research, led by Adam Cohen, investigated the ability to analyze a plethora of different measures, including digital measures, with their Trial at Home platform in a condition, a rare disease, that has intellectual disability as a core feature. But the suite of measures that they look at are incredibly relevant to autism. And their whole pitch was that it's never going to be one measure, one biomarker, or one endpoint. You really need to look at the totality of this and utilize these digital tools to have much more accuracy and precision in understanding what's going on, particularly in social and intellectual disabilities. So I'll bring this to your attention because I know they're very excited to go beyond the few ends they looked at. In this case, they might be a great contact to work on for those interested in this rapidly evolving area. So in summary, I'll just share with you that I'm so excited to see the progress that's occurred in autism since 10 years ago, since I worked in this area. I do believe that CPath Institute, we're growing so fast. We now have over 120 staff. We have really exciting, talented people with core competencies that could really help advance the progress that's already been made, particularly bringing, today I talked to you about biomarkers, but we have a lot of success looking at modeling tools. Last week's presentation by the Roche team, we actually have a success in building modeling tools that can help predict what the placebo response will be based on data. We've done this in Alzheimer's, and it'd be so amazing in autism to be able to predict what the placebo response would be in any trial. Growing efforts in pediatrics, we have a generously funded grant from FDA to work on rare disease, clinical trial simulators, and I mentioned the digital health. So I know I said a lot, but I'm happy to take any questions, and I want to thank and acknowledge all the CPath staff, our funders, and particularly the partnerships and alliances we have with FDA and EMA.
Thanks so much, Diane. Super exciting what you guys are doing and the traction you guys have made in really catalyzing advancements in Parkinson's and other areas. So does CPath have an active or planned program for autism?
We don't at this time. Several years ago, I interacted with friends that ended up going from Pfizer to Autism Speak that are no longer there, but we don't have an active autism program at this point. We do have a rare disease initiative funded by FDA that has organizations such as the Rett Foundation, TSC Alliance, and others that might have interest in an effort looking at the monogenic forms.
Okay. From a community perspective, what would we need to do to engage with CPath to assess their interest for autism?
Yeah, I mean, what's so exciting is many of the folks that are on your speaker list just today and over the series are people that we have interacted with, the FNIH and others. So it would be great to just set up a call and talk about, what are the areas that we already have core competencies in where there's gaps or other opportunities. We work very closely with the FDA, Office of Neuroscience as well. So maybe just engaging to see where are the gaps and what are the things that we could do to help fill the tremendous progress that's already happening.
Okay, excellent. Let's see, there's another question. There are ASD subsets that have movement disorders and hippocampal abnormalities, sclerosis, for example. Do you see your work in Parkinson's and Alzheimer's as a springboard for ASD?
That's a great question. I haven't really followed the neuroimaging literature or findings in research in autism of the recent past. We are bringing on some expertise in neuroimaging to join us at CPath at the beginning of the year. But, I haven't actually explored that, and I'd be really interested to learn more about which type of imaging modalities. I know there's a lot of functional imaging modalities as well, fMRI and others might be of interest to consider a regulatory path.
Okay, thank you. And I think this will be the last question. If you had one wish or could wave a magic wand, people that are developing either biomarkers or therapeutics for autism, what would you, Diane Stephenson, recommend to them as your one wish?
That's a great question. I would say the biggest wish I have came up in the question to the previous speaker. We need to figure out how to incentivize data sharing and to alleviate the risks and the concerns. Because if you talk to parents of children with autism or, the patient community, the patients really want to share their data, but it is the most difficult thing we do. Once we get the data, the magic can happen. It's just the hardest thing we do. And it's really a challenge in the digital health space. It's just this competitiveness or concern for IP or confidentiality or things I'm still trying to figure out. But that's the biggest gap to go from where we are to where we need to be is to expand data sharing.